Okay, for the third time we're going to try to do this and avoid a virus. We're going to go to index B, which we can just hit the index here, look for index B, and then go to bacteria flagellum, refuted. Okay, now we're going to look for where we left off. ID enthusiast. Okay. Now, we're talking about signals which must be sent out while the components have not yet evolved by chance, in which case nothing would be accomplished yet. These are some signals from the bacterium. Then nothing would be accomplished, but the other two things would have been accomplished. But you don't get all three together. That's talking about as we move up here. If one gets the sequence of all the proteins right, localization, in other words, in the proximity of the species, and then the assembly instructions are carried out, this evolutionist maintains the localization and assembly will take care of themselves. Not necessarily. The protein sequence may be arriving correctly, but there's not the right location, and right assembly instructions may not be sent out. Well, they may be sent out for assembly, but the components have not yet evolved in the position of local and assembly and the right proteins. So nothing is accomplished, and we have to go starting over again, and many more years go by. Now he maintains, however, that to the ID enthusiast, however, this is a point of little concern. According to Dembski's, evolution could still not construct the 30 proteins needed for the flagellum. His reason is that the probability of their assembly falls below what he terms the universal probability bound. According to Dembski, the probability bound is a sensible allowance for the fact that highly improbable events do occur from time to time in nature. The probability depends whether it's successful or not. Depends upon the amount of time that goes by. If it goes too much time, the universe is gone and gone. To allow for such events, he agrees that given enough time, any event with a probability larger than 10-150 might well take place. But there, if a sequence of events such as a presumed evolutionary pathway has a calculated probability less than 10-150, we may conclude that the pathway is impossible. If the calculated probability is greater than 10-150, it's possible, but unlikely. These are just speculatory things. From either point of view, we don't have really definitive proof either way. We have to look at the preponderance of the evidence, because we can't observe, repeat, and falsify. These three things. Not science. But what does the preponderance of the evidence show? Well, let's go on with Dembski. Dembski turns his attention to the chances of evolving the 30 proteins of the bacterial flagellum. He makes what he regards as the generous assumption. Guessing that each of the proteins of the flagellum have about 300 amino acids, one might calculate that the chances of getting just one such protein to assemble from random evolutionary processes would be 20-300, since there are 20 amino acids specified by the genetic code. Dembski, however, concedes that proteins need not get the exact amino acid sequence right in order to be functional, so he cuts the odds to just 20-30. That's too far. Which he tells his readers on the order of 10-39. He hasn't done enough homework here, it seems. Since the flagellum requires 30 such proteins, he explains that 30 such possibilities will all need to be multiplied against each other to form the origination probability. That would give us an origination probability for the flagellum of 10-1170, far below the universal probability bound. The flagellum couldn't have evolved, and now we have the numbers to prove it, right? Well, suppose the time frame for this exceeds the proposed age of the universe, just for this one simple life form, and then you add all the others, mammals and man. Wow, we have too much time for the universe to be as it is today. Assuming impossibility. Well, this professor who goes against Dembski, I have no doubt that to the casual reader, a quick glance over the pages of numbers and symbols in Dembski's books is impressive, if not downright intimidating. Nonetheless, the way in which he calculates the probability of an evolutionary origin for the flagellum shows how little biology actually stands behind those numbers. And those numbers are too speculative at this point. We need Dembski to do a little better work. His computation calculates only the probability of spontaneous random assembly for each of the proteins of the flagellum. You combine them together, all occurring at the same time in order for the component to work, then you got too much time. Having come up with a probability value on the order of 10-1170, he assures us that he has shown the flagellum to be unevolvable. This conclusion, of course, fits comfortably with his views is that the Darwinian mechanism is powerless to produce irreducibly complex systems. Okay, you can't do it random, it has to be intelligent designer. However, complex Dembski's analysis, the scientific problem with his calculation is almost too easy to spot. By treating the flagellum as a discrete combinatorial object, he has shown only that it is unlikely that the parts flagellum could assemble spontaneously. Unfortunately for his argument, no scientist has ever proposed that a flagellum or any other complex object evolved that way. You have to go into details. We can't observe, report, and falsify. So there's not enough time in our lifetimes. And we have to have all this done over ages and ages of many human beings working together. So far, we have to do the best we can. And it looks like intelligent design still looks at this preponderance of task of this random processes of putting this hugely complex component together, whether it be locomotion or toxin exposure. Both of them are so unbelievably complex that we have to look to an intelligent designer until something better comes along. Dembski, therefore, has constructed a classic straw man and blown away with an irrelevant calculation. Well, it's in the right direction, but he's premature, let's say. By treating the flagellum as a discrete combinatorial object, he has assumed in his calculation that no subset of the 30 or so proteins of the flagellum could have biological activity. As we have already seen, this is wrong. Well, we don't know. Nearly a third of those proteins are closely related to components of the TTSS. That's apples and oranges. How did that evolve in the TTSS? See, which does indeed have biological activity. But how long did that take to evolve? A calculation that ignores the fact that it has no scientific validity. When we observe something and its components to the best ability, which both have done, and we say, well, one is similar to the other, but not the same, and the process by which one evolved, if it did at all, is not the same as the other. More importantly, Dembski's willingness to ignore the TTSS lays bare the underlying assumption of his entire approach towards the calculation of probabilities and the detection of design. He assumes what he is trying to prove. Well, he's pushing it. He's pushing it. He hasn't done enough. He hasn't admitted that these are only estimates, and how close can the estimates be? Sometimes you just have to say, well, this is so far, and the burden of proof is on the opposition to disprove at this point. And so far we have an intelligent design or preponderance of the evidence. According to Dembski, the detection of design requires that an object display complexity that could not be produced by what he calls natural causes. In order to do that, one must examine all the possibilities by which an object, like the flagellum, might have been generated naturally. We can't do that. It would take a whole lifetime to do that, and more. And random processes is so vast that's beyond the capacity of the entire population of the world if we were all scientists. Dembski and Behe, of course, come to the conclusion that there are no such natural causes. But how did they determine that? Well, that's subjective, yes. What is the scientific method used to support such a conclusion? It's educated guesswork, but poorly done. It's in the right direction, though. Could it be that their assertions of the lack of natural causes simply amount to an unsupported personal belief? Yes. But also, it's been investigated and we're going in the right direction. Suppose that there are such causes, but they simply happen not to think of them. That's true, but we're looking for something we haven't found. We can't estimate what it is that we haven't found. Dembski actually seems to realize that this is a serious problem. So does the evolutionists. He writes, now it can happen that we may not know enough to determine all the relevant chance hypotheses, which here as noted above all means all relevant natural processes, but we don't know what they all are because we don't know all of them. Alternatively, we might think we know the relevant chance hypotheses, but later discover that we missed a crucial one. Well, we're going to keep looking at things and looking at things we're not going to get to. It's virtually, for us, unlimited. In the one case, a design inference could not even get going. In the other, it would be mistaken. Well, we don't know that because we don't know all the possibilities. Let's just look at it and get it. The preponderance of the evidence favors intelligent design because we don't even have a whit of evidence in the direction how natural science, random processes could accomplish these two complex mechanisms in such a time that it would be actually existent in the universe. Maybe they're still going, and maybe in another 25 universes from now, they'll come to bore. But it doesn't look like they're going to come anytime soon, so let's stick with intelligent designer until that comes. What Dembski is telling us is that in order to detect design in a biological object, one must first come to the conclusion that the object could not have been produced by any relevant chance hypothesis, meaning naturally evolution. Then, and only then, are Dembski's calculations brought into play. Stated more bluntly, what this really means is that the method first involves assuming the absence of an evolutionary pathway leading to the object, followed by a calculation proving the impossibility of spontaneous assembly. Incredibly, this is a priori reasoning. This a priori reasoning is exactly the sort of logic upon which the new science of design has been constructed. Well, we're both in the same direction, evolutionists and creationists. We can't observe, falsify, and repeat. So we have to do our best, using the best scientific approach that we have, minutia points here and there to see where the preponderance evidence favors one or the other or neither. So far, it seems to favor intelligent design because it's an intelligent design. Now, how could it have occurred unintelligently, randomly? So far, we don't have a good plan that even comes close. Not surprisingly, scientific reviewers have not missed this point. Dembski's arguments have been repeatedly criticized on the issue of on this issue and on many others. Designing the cycle and assessing the design argument, it only seems as though two distinct arguments have been raised for the unevolvability of the flagellum. In reality, these two arguments, one invoking irreducible complexity and the other specified complex information, both depend upon a single scientifically insupportable position. That's true because it's not science. Observability, repeatability, falsifiability. See, and the irreducible complexity of an organism that is locomotion on a bacteria and the one that secretes toxins in another bacteria are 2 points, two different separate ideas. Both have to evolve the way they look. How long does it take each one? The two aren't indicated as coordinating with one another. Remember, random. They don't have a relationship with one or the other. It's not some kind of signal that one species is signaling to the other to evolve into something of a component for locomotion for the other, or the locomotion species is helping evolve the other species into a toxin release mechanism. Namely, that we can look at a complex biological object and determine with absolute certainty, we can't, that none of these components parts could have been first selected to perform other functions. Well, it could have. We see it. But one has nothing to do with the process. We have machines today that have same bolts and nuts and certain components. And we say this component here can be used for a number of things. But it's a different, separate machine. And the way that machine is assembled is different from the other machine. That's simple, basic logic. The discovery of extensive homologies between the type 3 secretory system and the flagellum are irrelevant. Has now just shown how wrong that position was. No, they can't. They're not related. They're similar, but not the same. They're not in the same function. Anyway, I answer, but you have actually observed, have you actually observed the bacterium and the locomotion component in which you replace that component with another functioning component that is similar or identical, found in another species that is similar to the first bacterium, to see if it functions properly either for locomotion or for other purposes such as producing toxins? No. Have you found any bacterium without a functioning locomotion component that's going to die because it doesn't have the wherewithal to locomote and get food? If you've not done any of these things, then your conclusions are pure speculation with no real basis for speculating, hence it's a waste of time. Note that an intelligent designer cannot be ruled out in any case because he could have created such functioning components as well as be received by the life forms he created. For just as he created sources of liquid and food to have it readily available for the life forms he created to utilize in order to sustain himself, so he could have created such components for the life